Okay, good morning. Well, it's a pleasure to be here. Thank you to the organizers for putting this session together. It's intended to be a quick view for people who are here today to see an overview of some powerful experimental techniques that are used for materials characterization that I'm brought to bear to learn more about cement and concrete. So back when I was a student at Northwestern 35 years ago, SEM, scanning electron microscopy, was the big, powerful tool. Everyone was starting to use it. It was really changing the way we thought about microstructure. But there was always a shortcoming of SEM, and that is it's a two-dimensional image. And you have to prepare the sample very carefully to get a decent image. Three-dimensional. How could we get a three-dimensional picture? Well, now we have it. X-ray computed tomography is emerging just as powerfully today for today's researchers to give a different view of the way microstructure is put together. A few acknowledgments about funding, but also those three key references. The first paper that I'm listing there is actually the one that sort of motivated this talk. Some researchers at Oregon State and our group got together to write a paper that was in Concrete International. And then below that you see two of our earliest papers on the work that you see here. So it's been 10 years already, but it's still emerging. People are still learning about computed tomography, and hence this talk. So scanning electron microscopy, two-dimensional, high resolution, and I show here some pictures from NIST. Paul Stutzman, the world's greatest electron microscopist. He did some fantastic work in the ACBM days. And then along comes X-ray computed tomography. And I found this older paper, might be one of the very earliest, 1980, where someone was using this to look at rebar and so on, very low res. By 1999, we're getting higher res. This picture is by my very good friend Eric Landis, who's in the audience with us. We'll give him a shout out. And this was a study where he was using X-ray CT synchrotron at a national laboratory where these samples were loaded, and his interest was to look at crack development. So you can see the crack there, and it was very high res, and it showed the progression and the character of that. So how does this work? X-ray CT, if you first think about what an X-ray is, it's a shadowgram. So if you get an X-ray on your broken arm, you'll see density differences, and you can see where the crack forms and so forth. Now the idea to get to computed tomography is quite simple. Now instead of one X-ray, do 180 of them, rotating the sample. So now we have this collection of images that we're going to give to a computer, and we're going to ask it the question, what structure are you imaging on all these different 180 images? You go figure it out, computer. And it works away, deconvolutes what is the most likely structure that explains all 180 images together. And that's what we come up with, a map of the microstructure in 3D. Many of the papers on X-ray CT still show two-dimensional style images, but they're really slices of the three-dimensional volume. So we'll see pictures like this, for example. This is a topic about in resolution. One of the sacrifices that we make compared to SEM is that X-ray CT is not as high of resolution. And we still need higher. And it's basically a drive of all these imaging techniques. Get better and better at resolving microstructure. And usually the way we describe resolution is how big is the pixel. And so you can imagine that that's not really the end of the story. Yes, we can see how big the pixel is in the digital volume, but the real resolution may be something different. We have fuzzy, unfocused images, and they're not highly resolving by pixel. So when you talk about resolution, it's actually a more complicated answer than just saying what the size of your pixel is. And resolution is important in all sorts of different imaging techniques. This is my nephew and his wife had a baby. And this shocked me when they shared this picture on Facebook. Look, that's a sonogram a couple months before birth of that little boy. And look at what's annotated up on his head. Hair. The sonogram could resolve hair on this baby. And so when he came out, we knew he was going to have a whole lot of hair. So in fact, that turned out to be true. And there he is, a little cutie pie. So now we have the lab-based remark. I wanted to say, you know, the high intensity, high flux source of X-rays is available at this national laboratories, synchrotron laboratories. But one problem is it's hard to get to a lab. It's hard to spend weeks and weeks at a national lab. And it was truly a breakthrough when the equipment has been made available for universities to get lab-based computed tomography equipment. It provides access and convenience. And even in that category, now the manufacturers of these lab-based units are seeking higher and higher resolution. We had micro CT, now we have nano CT, so trying very hard to impress on how much they can resolve. You can see some numbers there. They've gone from conventional medical CT millimeter scale down to supposed nanometer scale. When you see at the very bottom line, X-rated nano CT has a spatial resolution of better than 50 nanometers, that's the kind of thing you want to a little bit take with a grain of salt, that can you really resolve your features that are of that scale. So we have a laboratory at, or a building at U of I called Beckman Institute, and they have these shared laboratories, and we have the micro CTs and the nano CT, and that is going to be the equipment that I use for this study. So we wanted to give this a try, and we thought first about being able to image interfaces. Interfaces in cement-based materials have always been of interest. We think about the diffusion of water through concrete and how the interface of the cement next to an aggregate is a very important feature of microstructure to understand. How does water move through this structure? So we made up our own interface with a little gold foil squeezed between the microscope slides, and we were making this sample so that we would be able to image an interface. And the next part, so there's a better picture of what we did. The better part is here. Now we're inside this, the nano CT. You can see it's on a stage. The stage rotates. The sample itself, if you're interested in doing things related to hydration, you have to ask yourself, how am I going to keep it wet? And so you need to seal it inside a vessel that keeps the evaporation from taking place. And so you can see that orangish kind of vessel there where the sample is located. And now the next thing is you want to do time studies that are at 1 day, 3 days, 7 days. So you need a way to put your sample back in place exactly where it was so you can take another image later in its hydration cycle. And so that's where the stage design, so you can lock it back in, is important. And so other ways of doing small samples are shown here. You could do small samples in a tube, etc. But just to quickly show you what we could get out of this, the microstructure is shown here. Different grains of cement are in the image here. And we have the 1 day, 3 day, 7 day, 28 day results where the sample is aligned with what in the time sequence. So you can look at something in the 1 day sample that is circled, that's a grain of cement. Look at the same spot in the later ages. You get a description of how it has dissolved and precipitated new product. So that particle basically goes away by the end of 28 days. There's another area that's empty at first day that fills in by 28. There's another Hadley grain kind of feature that you see here where it develops a new kind of low porosity zone around the particle, and so on. So we would now have this nice digital image of these of the same sample at different ages, and we're able to do 3 visualization of these kind of data sets. And we can just separate out the paste or separate out the pores, show the wall effect at the interface, and so on. And this helps us understand some of the things we knew about with SEM studies, but now can do studies in 3D. So if your question is about, say, diffusion, well, diffusion now can be analyzed as a 3-dimensional kind of function. Okay. So we can separate phases. If grayscale can be figured out to be our way of separating, then we can separate phases. And in the case of separating the pores from the solid, we can do that. There's the hydration products. Here are the anhydrous cement. Put them together, we have a multicolored solid. And you can imagine how this can be used for different kind of studies with diffusion, durability issues, and so forth. The main part of this talk, or the second part of this talk today, is to feature the CI paper, Concrete International paper we had with Oregon State researchers. And we included kind of an overview. It was basically trying to spur people's interest in X-ray CT. Here are the possibilities. And so we put together a couple examples. The first example was to feature cellular materials. We had been working at U of I on these lightweight cellular materials, and you can see there an SEM image on the left with the 3D CT reconstructed image on the right. And we've oriented them for these paired of pictures just right so that you can basically see in the 3D image what you're seeing in the 2D image from SEM. We wanted to do this as one of our very first things to build our confidence that the X-ray CT's reconstructed image was real. After all, it's just a function of a computer calculation. Is it real? The SEM gives you a little more confidence that what you're seeing is real. And this was actually consoling to us. We thought that match was pretty darn good. And we could get the same dimensions, the edges were in the right location, the line distances were in the right size, and so forth. So now we have a three-dimensional image that we trust, and now you can do other things with it. For example, you could use a 3D printer and go ahead and magnify the thing by 400 times and have a copy of your microstructure in nice yellow plastic that you can share and look and get a feel for what the microstructure looks like. You could also do calculations. You could do a three-dimensional volume size distribution of the bubbles that we had in those cellular structures. We could also take that same mesh and say, hey, that actually constitutes a finite element mesh. We could take the experiment from CT, make the mesh a mesh for finite element analysis, and load it. And we can, what you see in that, the image in the middle is sort of a stress intensity of how the microstructure is carrying load when there's an external load on it. This would be actually a little similar to what Eric's experiment was about. If we work those two techniques together, we could see where the computed high zones of high stress match up perhaps with where the cracks form. At least that'd be the speculation that they would. The second study of our paper in CI was this phase segmentation. This was a study by OSU. It was at the Advanced Photo Source at the Oregon National Laboratory. The first step for this experiment was to calibrate the grayscales. We're going to try to do phase segmentation based on grayscale, so let's go ahead and build a special sample that has those layers, and then we can get the grayscales to match up. And that's what you see on the top. And then by the lower two images, that's the microstructure where the grayscales, well, the color is mapping what kind of phase is being assigned to that. So once you have that, you could do a number of different kind of studies that might deal with durability issues or any other kind of fracture issues that you might be studying. A third study in that paper was monitoring deterioration. And this was kind of interesting. It was a proof of concept experiment. And it was basically, can we get faster imaging? The X-ray sources at the synchrotrons have a monochromator to keep your X-ray source a very defined frequency. And so we could take that out, compromise the quality of the X-ray source, get higher flux on the X-ray source, and get our images much, much faster. And so this technique increased the X-ray flux by 1,000 times, and thus reducing the time to make one image from 30 minutes down to 15 seconds. That creates a lot of possibilities about catching the dynamic process. So, and this is the pictures they had with an opal aggregate. So, in summary, X-ray CT is a powerful technique to visualize microstructure, pores and phases and interfaces and cracks. Resolution is adequate. We would love more and higher resolution. And I think through the time, I think we're going to see these machines improved. Lab-based X-ray CTs are showing up everywhere. They are truly convenient. University campuses are finding ways to get these things, and so you very well might have access to these kind of machines for your own studies. And last, the remark about lab-based CT brings these techniques to a college campus near you. And yeah, I forgot to mention this idea of doing wet studies. That certainly separates X-ray CT from SEM. SEM, you'll only get dry samples generally, unless you use an environmental SEM. So if you want to do things that are in a wet condition, the X-ray CT is a good option.